patients access molecular diagnostics to improve diagnosis, but also ensure that we do drug sensitivity testing, be able to curb the dangerous germs that could cause high mortality, and also ensure that the quality of the diagnostics is good. So as Uganda, just for us to appreciate the team that could bring you to this, we have a diagnostic network that is quite well supported, with microscopy being our backbone for a long time. But currently, we've been able to expand the molecular tools like GeneXpert, which are available in over 340 facilities, and these are also supporting our disease testing. We have the TrueNut portable tools. We have a TB lamp, and we are trying to scale up TB lamp in community testing. We have additional 33 that we plan to install this year. We have LF lamp for HIV, people living with HIV. And then we have invested in dict X-ray with AI for TB screening. Currently having 70 portable dict X-rays, and we are adding more 11 mobile clinics with 56 portable X-rays. Our target is to have every district have a portable dict X-ray that can support facility and the community screening. Then we are piloting AI for predicting TB hotspots, so that we can be able to put the communities with more information on where we expect to find TB. And then we also have culture labs that are doing culture and DST, but also have used connectivity, where we have in lab expert supporting monitoring all these equipments and improve transmission of results. So where are the gaps that we still have in the network? We still see that by the standard, 80% of our population should be living within a 5-kilometer radius to where we are a molecular diagnostic tool. Here we are at 30%, and of course this causes other delays, or issues with delays to access diagnosis, and this will lead to increase in catastrophic cost to TB disease. So this is one of the gaps that we need to improve, access to diagnostics. In other gaps, we see, of course, long result time. We know that from the data, 84% of our population are accessing molecular diagnosis. These are the new and relapsed patients, but some of these results come back after days or weeks, and sometimes can be even longer than that, especially when we go into community campaigns. So we think that issues to do with having tools that can be available closer to the patients and have a patient tested and put on treatment can help us to address TAT. Then an area with engaging the private sector. We know that we have thousands of private facilities where patients go, and from our data from TB, we see that over 50% of people with symptoms of TB first go to the private sector for support. But this is where we have a big gap in accessing diagnostics. So we need to see that we can have tools that can be accessible to the private sector, but also issues to do with the cost of diagnosis in terms of the investing in the equipment. Currently, all the available equipments are heavily expensive. The commodities are very expensive, but also issues to do with the service and maintenance. Not only do you need to have trained service technician from the companies, but also the cost of service is very high. So these are some of the gaps that we need to address, and these are things we need to we're currently working on so that we address. This is what I wanted to share. Chair, over.
Thank you so much, Abdul. Thank you so much, Prof. I think this is very good. It has set the pace. It has given us the status of our TB diagnostics and the gaps. So I think this was very good for that. Sorry for not seeing the message you had shared. So thank you so much. And over to you, Alfred, to make your presentation. Alfred, over to you.
He's trying to unmute. Yeah, click on that, unmute. There you go.
Can you hear me now?
We can hear you, but we can't see your presentation.
Oh, just a moment, please. Can you hear me? Can you see the slides now?
No, we are not seeing
On
your
my
slide.
side,
We are
no.
seeing Dr. Susan's iPad.
You're now on.
Alfred, do you mind sharing with me and I share from this side if you have issues on this side? It's now on. You just need to put in slide mode.
Yeah,
Good.
I think it should be now. So sorry about that. Good morning, ladies and gentlemen. Thanks, Abdul Nur, for organizing this, and also Susan and all the rest for giving me the platform to share done over the last couple of months that has resulted into this presentation. I would like to really acknowledge all the partners in this, including my colleagues who are also attending right now. We worked with the different sites in achieving all the data that some of you must have already accessed. Just to add on to what Abdul Nur just presented, we know there's been a gap in the diagnostics. Although WHO recommended several molecular-based tests to penetrate the lower health facilities, this has not been achieved for those reasons that Abdul Nur cited, from infrastructure, cost of maintenance and distribution and all of that. Over time, WHO has been recommending tests that can actually be placed at the community level to try to address the issues related to notification of TB cases and starting them on treatment, effective treatment, and also stop transmission. Data available from last year's WHO report indicated that only 48% had access, and of those, less than 50% had actually access to those recommended tests. Now, as we are here in Mulago, in Uganda, we have been operating under the Rapid Research for Diagnostic Development. The goal of this particular study is to advance the next generation of TB diagnostics, especially in the areas where these tests are expected to be used. There are several objectives for this study, but how it operates is that we work with the different product developers. These include early prototypes or late or designed locked devices. The idea is to evaluate these devices in the real world situation to assess their accuracy,
I want
their
to send you this
usability.
on the side that you join.
Sorry. Yeah, so the aim is to reach out to these different developers. Evidence is that once we have evidence involved data generated from research activities, it takes usually not less than 15 years for them to come to practice. So in order to shorten this period, we've scouts that move around the world, identify developers, and profile all these devices to see which site they will fit best. So for the devices that we have evaluated here in Uganda, these are mainly meant for sub-Saharan Africa, where there's high TB burden. So the study design is similar across all sites. For this particular data that I'm going to share, the study was done in Uganda, Vietnam, and in India. And in this study, we enroll consecutive people more than 12 years. And we then explain code for at least 2 weeks and have other risk factors, including HIV, working in the mining industry, and also those who have contacts with the TB patients. And we excluded all those who are on active treatment and all are taking medication of antimicrobacterial activity or are unwilling to participate in the studies. We also limited participation in a 20 kilometer radius from the site of enrollment. This facilitates return to follow-up visits and also be able to call them quickly to come back and initiate treatment if they tested positive. And so the aim of this study was to compare the index set, which is the Plus Life, and MolBio on tank swab and sputum, together with the routinely used tests, including the GeneXpert Ultra and smear microscopy, with liquid culture as the reference standard. So at all the enrollment sites, in this case, we have Mulago TB ward, we have MJAP at the ISS clinic, and Kiseni Health Center 4. So at each of these sites, we do chest X-ray for patients, all the patients, and we collect sputum. We collect blood to do HIV testing and the HbA1c for ruling out diabetes. Then we collect tank swab prior to sputum collection. And this we do it by rolling the swab on the tongue of these patients for about 15 to 30 seconds. And if the provided sputum, we'll swirl the swab in the sputum sample before transferring them into proprietary buffer. Now, like I mentioned, we had a definition for our reference standard. This included a positive liquid culture. And if the two cultures came out negative, then that was a negative test. And there are results that were categorized as intermediate if one culture was contaminated and then the other one was negative. So for the comparator test, like I said, we used the GeneXpert Ultra. Then we also did the sputum microscopy using Oramine. For the Ugandan, like I already mentioned this, so these patients are normally enrolled at the gates. We have a study team stationed at these gates. If you came to Mulago or to Kiseni, you will find a table there where we have a stationed either social worker or nurse, asks the patient on the eligibility, especially for cough, at least 2 weeks. Then from there, they take on the patients to explore more about the symptoms, and if they're concerned, then enroll them onto the study. Initially, we were doing all this here, and then the testing was spread all over the hill of Mulago, including Ward 516, pharmacology, and sometimes in the pathology building here. Now, I just wanted to give you a brief history about the Kebera Diagnostic Laboratories, where we have been doing all these tests and where we have set infrastructure to evaluate different diagnostics. So we are located at Uganda Nurses and Midwives Union premises, just outside the Mulago main gate. And this idea of setting up this lab started when I was a PhD student. being supervised by Professor Joloba Moses and the Professor William Worodria. And in the 20,
Can you
Abdunur,
mute Sarah? Or can you mute people?
thank you, Susan. Thank you, Abdunur, for making sure, by Global Health Labs to, we have been working with the Global Health Labs. This is part of the labs into innovation at Gates Foundation. So we have been working with them since 2014. So in 2022, they invited me to talk about to share with them about our collective research and what perspectives I have for Uganda specific and what we could do in order to become self-sustaining. So I took up that opportunity to tell them that, in case the funding from their end dries up, what would we do as a country? And they've helped us build this capacity in terms of training over time. So what would we do with all these people who have been, really putting all the energy in realizing all these innovations? So they encourage us to build something local, give it a local name, and operate it in the local context, and make it open to whoever is willing to work with us at our own terms. And that's how we looked at Kebera, and we brainstormed with the two of my colleagues who are also on here, Talema and Lucy. I really appreciate all your brainstorming during those initial stages. So currently, we are undergoing quality management system towards accreditation, and we are being guided by Mr. Patrick Ademoun. We hope this comes through before the end of the year. So in terms of the laboratory, we have about 9 lab technicians and technologists who are highly trained and motivated to do their work. We have array of different products from different manufacturers that we're evaluating. We have a stationed biosafety system that is highly maintained. Then we have additional infrastructure that supports all the investigations that we do on these patients. And we have a very strong, standby generator that's in case power goes off. We are really covered along that line. So in the lab, we have the basic testing platforms like the GeneXpert 10 color. We have the microscopy. We have a Quanti Studio 0. Then the mini doc and the duo, and we also have the MolBio TrueNut, which we also test concurrently. So since for the first 5 years, we have evaluated about 21 different platforms, and out of these, the one that has come out clearly that we're actually proud of is the Plus Life. We have others whose data we're trying to compile and make available to different partners. And the most recent, which I've not included in this slide, is the NATOS. NATOS is a nucleic acid test on a strip. So it's like a dry strip, like for malaria RDT, which we're evaluating. We're hoping by the end of this year, the results will be available. Before that, we got the Coyote. All these are molecular based. And I think they try to work in such a way that the devices can become closer to the patients who need the test. Now, let me talk about the Plus Life Minidoc MTB. The picture you see on this slide is exactly how it looks like, and it has the strip which is inserted, as you can see it here. Using the same approach of enrolling patients, the same patients that we evaluated this particular Plus Life machine on. It is a very simple machine, and it's portable. Just can hold it, can't actually fill the whole of your palm. And like I already explained, we collect tongue swabs or with sputum swabs from the cups. We mix them in a proprietary buffer. Then we lyse them using an automated machine. Then we pick about 400 microliters, put it in the card. Then that card is inserted into the plus life. Then within 30 seconds, 30 minutes, you should be able to get your result. In fact, our experience in the lab is that sometimes in positive cases, even below 20 minutes, you get your result. Actually, the range is usually between 15 and 18 minutes. So, but the upper end is 30 minutes. So this significantly cuts on the turnaround time. And as you can see, that mini doc alone, the throughput is about 9 samples in a day. So if you have like 2, and with these sites that have maybe about 10 or so on, actually you can manage this within the setup of this. This is according to our own experience. So the principle of this test is that it employs isothermal amplification, unlike the usual PCR where temperatures vary according to the particular aspect of that assay. This maintains a constant temperature, and then the changes in the detection and amplification occur at the same temperature. Then further, the sensitivity is enhanced by the thermolyze, which automates and heats the sample, and then by sonication makes the DNA available to detection. Now, what did we find? So the data that you see here was what we got from our lab here. First of all, the error rate, especially for tongue swab, was so low. Although it was lower than the one for sputum swab, this was not really significant in terms of context to use. Then the sensitivity for the tongue swab alone was 85%, compared to sputum swab, which was at 89. Then in either case, the sensitivity was so high. Now, compared to the comparator test, that's sputum expert and microscopy, you'll find that the tongue swab performed much better than the sputum smear, which was about 67%. Then compared to the sputum expert, which was at 94%. Of course, sputum is known to be superior to tongue swab. But what the plus life will bring on the board is additional yield, because many people will get tested using this tongue swab, or if they give sputum, then the sputum swab. Then we went ahead to look at cost between a sputum expert and tongue swab testing. And we found that the cost of testing using the plus life on tongue swab or sputum swab will be less than half of that of gene expert. That's Uganda specific data. Then, because based on that data, we conclude that the Minidoc and the Ultima from AllBio have exceeded the WHO minimum accurate threshold for both non-sputum and sputum-based point of care. And sputum swabs had similar accuracy to sputum expert ultra. And both tongue swab and sputum swab tests outperform smear microscopy, which is actually the stay and takes a large proportion of diagnostics in this country. So swab-based testing, therefore, could increase access to rapid diagnostic tests for all people with the presumed problems of TB. And would test sputum as well to increase sensitivity, and tongue swabs to maximize yield. So, therefore, we recommended that multi-country clinical sites could be recommended in order to guide WHO guideline development group. I think we have provided this data to WHO already. We are expecting that in November there will be a meeting to take decisions on how this could fit in that context. We are soon initiating usability studies in our sites, especially here in Uganda. We are awaiting IRB approval to assess how the users, including the nurses, those who will test in the field, and also clinicians in terms of accepting the results of these tests in clinical care. Then we also think further studies are required to to explore performance in key subgroups and in the context of active case finding. Now, we sat back and thought about what else could be required to influence uptake of this particular test. We looked at logistical requirements, which include the cost. And for your information, the cost of the test, as you saw, is $3 or less. And the device itself are like about $150. Training. Our experience is that training is very simple. If you took, let's say, half a day to train a caregiver on how to use this platform, they can actually, less than half a day, they can be able to use it. After all, at village health teams, they're able to use BP machines, glucometers, and other TTs. So processing is so simple. Like Abdul, maybe some noise. I don't know whether you guys are able to hear me.
Sarah or Shamim, can you mute all those that are making noise? I mean, that whose mics are open.
Thank you, Susan. So processing a sample for this test is quite simple. Like I already explained, you get a swab, put it in a buffer, then you swirl it, and then thereafter you take a portion of that sample, put it in the card, and within 30 minutes you have your result. So the earlier version did not have a battery backup system. We recommended to the developer, and they thought this was something simple to do. So once that is done, I think it becomes handy even to be deployed at the community level. Throughout the study period, we never had any maintenance issues. As if you can remember, the error rates were very, very low. In fact, Ugandan site had like 100% of detected results out. Then, yeah, so the other question we asked the manufacturer is whether this particular device can have an advantage over the other tests. For example, can they do multiplexing, have different age groups, have different specimens, have various infectious diseases detected on the same platform? I think these are things they are probably now working on to incorporate. So for Uganda, I don't have any clear answer, but I think this answer may come from this particular meeting. Initially, we had the discussions with the Prof. Joloba and the National TB Program to see if we can do a demonstration study. This was aimed at seeing how this particular platform could fit within the current diagnostic network, and also whether we can bring other partners, including private sector, because we know, like Abdul Nur already pointed, that a large proportion of our lower health facilities have 130, over 1,000 microscopes, and all these feed into a hub system, which hub system becomes expensive in terms of turnaround time, making sure these riders, are able to deliver in good time, take in a good time. And if you went to our health facilities, you find a lot of unclaimed results, some of which are positive high on expert, and these patients are continuously spreading TB disease. So these are questions we need to answer. And the big question is, can the results of this mini-doc plus life be
Alfred, you are muted. Alfred, you are muted.
I think Sarah muted everyone whose mic was on.
Alfred, you are muted. Please unmute yourself and continue. Okay.
Yeah, actually, I was almost done. So I was just acknowledging the three the study teams that we worked with in generating all the data that I've shared. Again, thank you so much, Susan, and your team for giving me audience. We look forward to the next steps jointly with you, and we are happy. Maybe one thing I want to say before I hand over to the chair is that the infrastructure that we have at Cabera Diagnostics can help the National TB Program, the Ministry of Health, in evaluating tests that might come your way. Currently, we are focused more on TB, but as you saw from one of those first pictures, we have also been able to work with the AI health lab at Makerere on developing an app for malaria diagnostics and the TB. So we could be as flexible as possible, given that we are setting up an infrastructure that's cross-cutting. I thank you so much, and for listening. I'm happy to take any questions through the chair, and we can discuss what next we can do in order to roll this to the people who need it most. Thank you.
Thank you so much, Alfred, for that detailed presentation and clearly letting us know how the machine works. So colleagues, you've heard, and from the communication already I've seen some of the funders like Global Fund with a lot of interest in using this machine in trying to pilot it out and the like. But we needed to understand because it was done in our country. One of the sites was our country. So here we are with all that information. Any questions, comments, clarifications to Alfred and Prof. Joloba, who was the team leading this? Yes, Ida, I see your hand is up. Please, you raise your hand if you can't type your question in the chat.
Thank you so much, Commissioner. Good morning, team. I just was requesting if Prof can, or Dr. Alfred can comment about the information system that is related to the Plus Life. Like he alluded to that, it was also put in the Chisenyi Health Facility. How were they connecting it to the available systems, or was it done? And if they also tried to troubleshoot on which kind of information system is compatible with it? Thank you, and over.
Thank you. Can we have all the questions come through, then we can answer? Hello? Okay, I don't see any other question. You seem to be content with what he has presented. Alfred or Prof. Joloba, over to you for that.
Yeah, thanks, Susan. Thanks, Ida. Probably Prof. Joloba will come after this. Yeah, so this study was being done in the context of research. And since it was investigational, we couldn't actually make these results available to the clinicians managing patients. And so the data that we captured was into our database. So it was not in the systems that are commonly used for remitting data into the other systems. I don't know whether either that answers your question. Of course, once it becomes a public utility, then it must be integrated in the current systems.
Yeah, I think either you are spot on that these are the issues we really we need to understand in the next phase from this demonstration research into now getting into real patient care and management. And unlike the other equipment we have been having, which are digitalized in terms of information transfer, this one also presents that challenge. It's of course more accessible, cheaper, faster, but then it is not yet with the connectivity solution. So we really need to go back into paper-based, figure out how to connect. But of course, it is like any other. There will be room for improvement. Of course, the things keep improving with time. So there are advantages and disadvantages, which we will need to navigate to put the system into play. But definitely, there is no doubt the advantages outweigh the disadvantages. If it continues to perform as the research has shown. Thank you.
Okay. Any other question or comment?
They're in the chat box, Chair.
Okay. Sorry. The questions requesting to share the presentation, then why 12 years and above? Andama, please take note. Why pilot was only done in Kampala? Then what is the method, limit of detection? How possible to access commodities for the platform and maintenance? Alfred and Prof.
Yeah, so let me also go first. About sharing the presentation, I can speak to Abdul Nur. Then we'll find a way of getting to the audience who need it. Then, in terms of the study location, yes, we did it mainly in Kampala for different reasons. Our currently the design was in such a way that we needed to have these results quickly, and also the reference testing within our reach. And but looking at the definition of a presumptive TB person in terms of what national TB program has stipulated, a prescribed period. So we had to do it that way in order to generate the result in that context. And that's why we thought if we were to go a demonstration study, then we needed to move outside these usual setups that we have here, and that will be selected on the feasibility of being able to do this within the time frame that will be allowed to do that particular approach of the study. Then in terms of maintenance, like I said, we did not have, we didn't need to do any maintenance at all with this particular platform. From the time we started using them up to when we ended the study and up to now, we are doing additional studies. We have never reached out to the manufacturer that it has malfunctioned, we need a spare, we need this. So long as you follow the procedures, and you know the way it is built, I guess even if it was in an environment where it goes like environment that is dusty, you can hardly see which side the dust will penetrate, probably through the port you use for inserting the card. Otherwise, and when it is running, you close it completely, such that you do not, have dust accumulate in it into it. Then the other question was commodities. That question may be beyond me. I think the manufacturer is aware, and the supply chain systems within the ministry or national TB program could probably work with the manufacturer to be able to satisfy any markets that might arise. Professor Joloba, you might need to add something.
No, I think the people, the questions they are asking, many of those are pertinent questions. And actually, that's why in the next steps, we are going into the early implementation phase. It's so attractive, really, to speed up this device. For the people in the field, they understand the gaps. Like Abdul has shown that we are struggling with the things we have. We are not meeting the indicators. We are not meeting the targets. We can't sit and wait. And this is really promising to give us an alternative. That's why it's being moved, but still doesn't mean that we jump steps. So that's why we want to go to our implementation phase, to work out those commodities, data management, performance. I've always taught in class that if you have something, you start in the best place. If you are testing a test, you don't go to begin to Karamoja. You start in Kampala, where things are more likely to succeed. If you fail in Kampala, then you don't waste going to Karamoja. So that is exactly the principle, that we start here. If it works, then we ask, will it work in Karamoja? Will it work in Bundibujo? So those are the next phases. These are people we saw who were coming, who were in the clinic, meaning they were sick. But TB now, we know we have asymptomatic people who have no symptoms seated at home. That's in the community screenings. Those questions we'll have to ask. Will this device, how will it perform in those type of people? So you can realize that there are many questions. Somebody is wondering about 12 years. These devices plus medicines, the process of bringing them to the market is usually long. 12 years might be even very short. It can be 20, 50. So investments in new devices or drugs is really a long-term commitment. So we are glad that this one has worked. We have been evaluating hundreds and hundreds of these. We're really happy that this one, evaluated also in Uganda, has worked. So like we have been lucky for many of these diagnostics which have come to the market, have come through our hands. Thank you very much.
Just to add to Prof, the other thing we also do in a similar vein is first testing any devices in adults. Then thereafter we roll them to pediatrics. So as we talk now, based on these gains, we started a recent study in children. Hopefully by mid next year or so, we will also have data to share, especially in a pediatric specific population. Thanks. Back to Chair.
Thank you so much, Alfred and Prof, for the updates and the information. So, Prof, kindly give us now a way forward in line with what's at level the ministry program and then the global direction in line with Plus Life, so that we know the next steps. Over to you, Prof.
Yeah, thank you. And I think, again, I'm very happy with this congregation today. They are spot on. The next steps is really, if as things go well, for us to go in early implementation phase, and we putting up together a team with all the people who are able and willing to really brainstorm which questions are key, what should the early implementation phase address according to us, and then we see, we test it out, and we have I don't like fingers crossed, but we will be on our knees that it performs very well, so that we relieve our population of this problem, contribute to solving this TB problem. So those are the next steps. So we have been invited to sit on the table of trying to see the early implementers, and hopefully we will remain there and then benefit. So as a government, we really look forward to government support, which they have already committed to really roll out this thing in Uganda and learn lessons and share with others. Thank you very much for coming and attending and showing interest. Thank you, Alfred, and your team. Keep up the good work. Thank you very much.
Thank you so much, Prof. and team, for always leading the way in TB diagnostics and innovations. And we look forward for that meeting where we've been invited as Uganda to be part of the early implementers to inform the next steps. So we look forward, and the findings and updates from that meeting will be shared to the team, and let us all be receptive as we start the early implementation. Thank you so much. And with that, I want to thank you for attending and wish you a lovely day. With that, I officially close our meeting. Thank you.
Thank you so much. Yeah.
Now and the next to come, medical laboratory is here to manage disease outbreak through science and through research. Medical laboratory, wonderful profession, the core of medicine.